Minindu. Hi. Hello. Can you hear me, Minindu? Can you hear me? Can you hear me, Minindu? Your microphone is switched off. Ah, yes. Okay. So if you can turn on the microscope and we can talk a little bit. Can you please switch on the microphone? Minindu, I can't hear you.
Miss, I can hear you,
Ah, yes.
but I can't hear it properly.
Now how is it? When I'm speaking loudly, how is it?
Uh, yes, Miss, I can hear now.
Yeah, it's okay. If you have a signal problem, you can switch off the microphone, then it will be fine. And so today we are going to start our grade 11 science classroom. So today is the first day, so I'm going to tell you something about me at first. So I'm Sanumali Jayakody, English medium science teacher. I'm a graduate of University of Sri Jayawardenepura, and I completed my degree, and now I'm working as a teacher. So, um, so I hope that we can work better to do our O-level exam well. So today, before starting the lesson, can you tell me something about you? I know your name, it's Minindo. It's fine. And tell me about what's your school and all, like those things. Hello, Minindo. Can you hear me? Yeah, fine.
Miss, my school is Carey College,
Yeah,
and I'm from Piliandal.
Piliandala. Yeah, it's good. That's fine. So today we are going to start our first lesson. I have to tell you something at first. In this platform, we are giving a tutorial together with our classroom. So I have uploaded that before the class. Um, since I am uploading it, normally I try to upload it a day before the class. Then you can take a printout and participate to the class. And at the end of the lesson, or most probably at the end of the day, we are giving a tute. It's like it may be a Google form, or it may be a paper. Then you have to answer it, and if it's a paper, you have to upload the answers back. So that is the normal process we are doing here. So Mininda, we are going to start the first lesson of grade 11. So I'm moving to the lesson. So I'm going to share my screen with you. Yeah, today our first lesson is living tissues. Now we are in grade 11 and we know what is the life is. Do you have some little idea about living things? Actually, can you tell me at first what are the living things you know? What are the things, what are the parts you can divide all the living things into? Tell me, there are three parts of this, all the living beings in this nature. What are those things?
I don't know, Miss.
Hello, teacher. What's your problem?
Yeah, now it's fine. All were muted. Now it's fine.
All right.
Thank you
Go
so
ahead.
much. Okay, Minindo, there was a little problem. So now we are back in the lesson. So we were about to discuss about the framework, and I was going to share my. Oh, again, there's a problem. Give me a second. I have to. Minidu, there was a problem with the control. So now we are back in the class. So we were about to discuss about the living being. So due to the delay, I'm moving to the lesson. Today lesson is living tissues. We are going to learn about the tissues. Uh, all the living beings are divided into main three groups. So they are plants, animals, and microorganisms. In case of microorganisms, there's only one cells. One cell. They don't have tissues. So we are talking about plants and animals and their tissues in this lesson. So here you can see there are many images containing plant tissues and different animal tissues. So first of all, I'm going to yes, first of all, we have to think actually what is a tissue is. So we have a definition for that. We have to remember that what is the tissue is. This is the definition for a tissue. It's a group of cells. That means it has some little amount of cells in it. And then with a common origin. They have origin from a common place. So we are going to learn this, what are those common place later on this lesson. So then it says that has been modified to perform a particular function in the body. It has a common origin, but now it has been modified to perform a particular function in the body. So as an example, in this image you can see this is a cross section of a dicot root. Here you can see there are some little squares and circles and all the things. So all of these are cells. These cells in different areas, those which are highlighted by these lines, they are having a same shape and they are arranged together. So these tissues are performing different specific functions in a plant stem. So later on we are going to discuss about what are those different tissues in a plant. So this is just an overview of what is a tissue is. Then it's a group of cells with a common origin that has been modified to perform a particular function in the body. So, Minindo, I have to ask a question from you. Do you have the Minindo, do you have the hard copy with you? Did you take a print out?
Sorry, miss what?
Do you have the hard copy of the tute which I have sent you?
I didn't get that copy or something. Miss. Didn't get the tune.
Yes. There's a tutorial. So in next class, you have to take the hard copy of that printout, hard copy of that tutorial before you are coming to the class. Okay. So reason is you have to fill some things in it. Okay.
Okay, miss.
Okay. Okay. So only for today, you have to take a printout at the end of tomorrow, and then you can fill the spaces with the help of the video, which is uploaded to the platform at the end of this class. Okay, that's fine. So today you don't have to write anything due to that reason. So you can involve with me together because you have to talk with me. Otherwise, you will feel that it is boring to stay without writing anything and without talking anything like in that way. Okay. Okay. Now we are moving to the next slide. Yeah. So plant tissues are divided into main two groups depending on their ability to divide. Those two groups are meristematic tissues and permanent tissues. So uh, these meristematic tissues and permanent tissues can be identified in a shoot tip or a root tip once we have taken a section. So this is in this diagram you can see what are the areas which contain permanent tissues and meristematic tissues. Here you can see this is the meristematic tissue area. It contains little small cells in it. And then in permanent tissue area, they have been changed into big cells and there are different colors you can see here. This blue color strip and this yellow color strip, they are the tissues which are doing a different function. And there's a root hair here, not root hairs. We can see here in this uh, lower region, which is having the meristematic function. So they do only division of cells. But this area contain the tissues which is doing different functions. So these meristematic tissues are again divided into main three groups. They are apical meristem, intercalary meristem, and lateral meristem. And the permanent tissues are divided into main two groups again. They are simple tissues and complex tissues. Simple tissues are again divided into three groups: parenchyma, collenchyma, and sclerenchyma. And then there are two types of complex tissues too. They are xylem tissues and phloem tissues. So this is the normal division, normal categorization of the plant tissues. Then we are moving to learn about meristematic tissues first. In this part, we are learning meristematic tissues at first. So here, at first, we are going to learn what are the features of these meristematic tissues. As I told you here, there are very small cells here. So in this image too, you can see that the meristematic region contain very small cells. So their most common feature is their ability to divide. They can divide and make another cells. And then these cells are not differentiated. The meaning of that is here in this image you can see in this area there's a blue color strip and there's a yellow color strip which is in this image. They are xylem tissues and phloem tissues, those which are performing different functions. And this root hair cell, it is performing another function. But the cells in this meristematic region, they are not differentiated. The only thing they can do is divide and make another cell. So they are really small size living cells. They are really small in size. And another feature is the presence of distinct nucleus in each cell. When we look at a cross section of a root tip or a shoot tip, in an enlarged diagram, we can see that they have a distinct nucleus. It can be seen very clearly. And then another special feature is they have large number of mitochondria because they want to divide very fast. Then they want more energy. So to produce more energy, they should have more mitochondria. And then another feature is no intercellular spaces. There's no intercellular space within each and every cell. All are packed together. If there is some intercellular spaces, we cannot see it very clearly. That's why we are telling that no intercellular spaces, or if there is, they are not prominent. And then they don't have a large central vacuole. Normally, we know that in a plant cell, we have learned in grade 8, in a plant cell, there's a specific feature that is a large central vacuole, but this is absent in meristematic tissues. There may be some small vacuoles in those cells, but there's no large central vacuole. And finally, no chloroplast. Their function is to divide, not to photosynthesize. So they don't have a chloroplast in their cell. Then these are the most common features of meristematic tissues. We have to remember those things. What are their features are. And then I'm going to discuss with you the classification of meristematic tissues. So here there are three types of meristematic tissues. As I told you at the beginning of the categorization of plant tissues, they are apical meristem, intercalary meristem, and lateral meristem. The apical meristem can be found at the tip of the roots or at the tip of the shoot or in axillary buds, we can find them. The function of this apical meristem is to increase the plant in height, if not to increase the length of the root, if not to increase the length of the axillary bud. It increases the length of that particular part. And then here you can see in this diagram, this is a shoot tip, this is a root tip. Both are growing further due to the activity of this apical meristem. It divides cells and it divides cells again and again. It make the length, increase the length of the root or shoot. And then I am moving to discuss about intercalary meristem. We can find them at nodes. The nodes can be normally found in monocot plants like sugarcane and also bamboo, those kind of plants. Sometimes areca nut. This kind of monocot plants have nodes. Here in this diagram you can see there's a node which is short in the first image and then it becomes long and long at the end due to the activity of the intercalary meristem at this node. They are dividing cells to upside and downside to increase the length of the node. And then that is a function of that. We can mainly find them in the grass family, another example. In grass family, we can see them clearly. And then the third type of meristematic tissue is lateral meristem. Lateral meristem is, it can be found laterally in the plant stem or roots. Normally, here in this transverse section of the shoot, you can see that there's a round circle. This circle is the meristematic tissue which is increasing the width of the plant. It increases the width of the plant. That means the diameter of the plant is increasing due to the activity of this lateral meristem. In a root, this tissue is named as cambium, which is mentioned here. It is cambium. We can see this diagram. There's a circle connecting all the vascular bundles in this image. These parts are vascular bundles. At the middle of a vascular bundle, we can see a cambium region. When the plant is growing further, this vascular bundle, the cambium tissues in this inside the vascular bundles come together and make a single circle, single ring, which increase the diameter of the plant. So that is the lateral meristem. Minindo, is these things clear for you?
Yes, Miss, it's clear.
Yeah. Is there anything you have to ask from me? Do you
Still
want
no.
to? Yeah, it's fine.
Yeah.
So, yeah, we are moving to the next part. It's all about meristematic tissues. Now we are moving to permanent tissues. Permanent tissues can be divided into two groups. As I told you earlier, they are simple and complex tissues. So the permanent tissues, the special feature of this permanent tissue is they have lost their ability to divide, and they are differentiated or they are specialized to perform a specific function. They are performing a specific function. In case of xylem, it performs a specific function. It has a unique function to do. And then phloem tissue, again, it is doing another function. In this way, they have a particular function. So simple tissue contain only one type of cells in their tissues, but complex tissues are having few different types of cells. So under the permanent tissue, we are going to discuss first about these parenchyma tissues. So simple permanent tissues, the first type is parenchyma tissues. They are living cells. They are isodiametric. Here, this word should be they have an isodiametric shape. If we can see them in a cross section, we have taken these cells are isodiametric. That means they have a round, that means circular shape, and they have a large central vacuole. Normally, in a plant cell, it's a common feature. Large central vacuole is present. And then, due to the presence of this large central vacuole, which is given here, this nucleus has moved to the periphery of the cell. It doesn't have enough space to be at the middle because there's a large central vacuole. Due to that reason, it is at the periphery of the cell. Then this thin wall, it has a very thin wall made up of cellulose. Just a thin wall made up of cellulose. And we can clearly see that there are many intercellular spaces within the cells. As mentioned here, there are many intercellular spaces within these isodiametric cells. Then, this is the normal feature of the simple permanent tissue, parenchyma. And then, these are some places we can find those parenchyma tissues. The main place we can find them is the pith of a plant stem or a plant root. Then another place is cortex. We can find them in cortex and in the pith at the middle. That is the middle region of a stem. That is one place. And then fleshy parts of the fruits. Here you can see that is a diagram of a tissue which contains some parenchyma cells. Here you can clearly see that there are some intercellular spaces in between those cells. This is given in red color, but you will think that what is the red color? So that's a fleshy part of a fruit. It is not photosynthesizing like a leaf. It has a vacuole which contains different pigments in it. So that's why it is red in color. Then mesophyll cells of leaves. Mesophyll cells, there are two types of mesophyll cells. They are palisade and spongy. This spongy mesophyll and palisade mesophyll. The mesophyll means the cells in the middle region of a leaf. And then here palisade parenchyma, spongy parenchyma, they are considered as mesophyll cells. So they are parenchyma cells. They have thin walls and they can photosynthesize because they have chlorophylls. We can clearly see here. And then the next example is seed endosperm. This is a diagram which contains a cross-section of a rice seed endosperm. You can see here cells with some intercellular spaces. And it is blue in color because it has pigmented with an indicator to identify clearly. And then we are moving to discuss about the functions of parenchyma cells. The first function which is mentioned here is the photosynthesis. As we see here, spongy mesophils and palisade mesophils carry out the photosynthesis to produce food in a plant. They are parenchyma cells. Here you can see the cytoplasmic streaming of the chloroplast in a normal plant cell. It's a mesophyll cell. The chloroplasts are moving here and there. They are looking for the sunlight in that way. So this function is photosynthesis. And then next function is the food storage. Here in tubers and in some fruits, the parenchyma cells are storing food. That is their next function. Mostly in yams and potato, we know that the potato tuber is made up of parenchyma cells. If we can take a section of a potato tuber and then we can color it with a particular pigment, we can put some iodine, because you know that in grade 10 in the first lesson we are learning that the starch granules, starch can be identified, can be easily seen with iodine solution. So if we can add a little drop of iodine to our section, we can clearly see those cells containing starch in a potato tuber. Those cells are parenchyma cells. So then next function is the storage of water in xerophytic plants. The most common example we know is aloe vera, because there's a very watery inside in a aloe vera leaf. If we can cut it, it has a gel-like thing. This contains mostly water. So it stores water in parenchyma cells. Not only aloe vera, cacti, all of these plants, all of xerophytic plants store water to withstand the dry condition in those areas. Then another function is provide mechanical support to herbaceous plants like balsam. Balsamiaceae family contain watery stems. They take the stem, that means the mechanical support to be erect by using these parenchyma cells. They store water in their vacuoles and the cells become turgid. Then okay, Minindu, can you hear me? I am asking a question. Minindu,
Yes, miss.
yes. Today I got to know that you were participating into another class. So I got a message from the control unit. So they said that you are going to start the class on 8:30. Is it?
What miss? Sorry, I can't hear you properly.
What is the reason?
Miss, can you tell it again? I didn't hear it properly.
Yeah. Yeah. Uh, I got a message from the control unit. They were saying that finish the class by 8:30. So is it?
Yes, miss. Because I was in the recording
Yeah.
while the time, and
Yeah.
now I have another class in 8:30.
Yeah. Yeah. I know. So is we have a little time left. So I want to finish some. Yeah, we do. We stop. then we are going to discuss about these simple permanent tissues. Here, collenchyma and sclerenchyma. We have another 10 minutes. I think we will be able to finish this area. Yeah, okay. Okay, these are the functions of parenchyma cells. And then we are moving to discuss about the collenchyma tissues. Collenchyma tissues are just like parenchyma, but they have a little difference in them. That is their shape and the nature of the thickening of the wall. They have a wall made up of cellulose, but there's a little difference. Their cell walls are thickened at the corners, just like in this image. And also they are polygonal in shape. As in the same way like parenchyma, they are living cells, vacuole is present, and intercellular spaces may present or not. Here, this collenchyma is considered as a modified parenchyma type, but here there's a little difference. So that is about collenchyma tissues. We can find them as a cylindrical tissue inner to the epidermis in the herbaceous stem. When we take a monocotyledonea, that means Vernonia. If we take a stem and cut a section and observe under microscope, we can clearly see there's a little layer like this which contain collenchyma tissues. That is the collenchyma tissue layer which gives a strength to that herbaceous plants, because we know that herbaceous plants doesn't contain wood in it. So it has to be strong. That function is given by collenchyma tissue. As in the same way, in this dicot leaf cross section, you can see that in this area and in this area, there's a little amount of collenchyma tissue. That's why we feel that veins of the dicot leaves are strong. That strength is given by these collenchyma tissues. And then I'm moving to see the places where they can be found. The place which I mentioned earlier, it provide the mechanical support to dicot stems before the formation of wood. Normally, not only in herbaceous plant, even in a plant which is going to be a plant which produce food, it also having some amount of collenchyma tissue in their seedling stage. And then it photosynthesizes in immature dicot stem. As an example, we can take this coleus tissue, coleus plant. We have these ornamental plants in our garden. This coleus stem is green in color. It can photosynthesize. It's an immature stem. And also, normally, in an each and every small seedling of a dicot stem, the stem is green in color at the very young age. They are doing photosynthesis. They are carrying out the photosynthesis. Here you can see these collenchyma cells. This white color, this glass-like parts, they are the thickened corners of the collenchyma cells. And then this is the third type of simple permanent tissues. It is sclerenchyma. Now we have learned about parenchyma, collenchyma, and then this is sclerenchyma, last type of simple permanent tissue. There are two types of sclerenchyma tissues. They are fibers and sclerates. The most special feature here is they are dead. They don't have nucleus, they don't have a cytoplasm, they don't have any other or cellular organelle. And also they are polygonal in a cross section. They have become dead cells because the chemical compound which is named as lignin is deposited evenly on the cellulose cell wall. Due to that reason, they have become dead. There's no pass of water, then uh, they cannot be alive. They don't have a nuclei. And then, uh, due to the presence of this thickening of the cellulose lignin cell wall, their central lumen or the pore at the middle of the cell, it had become a tiny lumen in sclerate modes of the time, and also even in fibers we can see a small lumen than other cells. Normally in parenchyma and collenchyma cells, we can see they are clearly live cells and they have a very thin wall. But here, the thick wall and a lumen is present. Then they are tightly packed without intercellular spaces. Normally, to produce the wood in a plant, they are becoming tightly packed and provide the mechanical strength to a plant. So, then where they can be found? Sclerate and fibers can be found in different places on the plant. The sclerate can be found in endocarp of a coconut or kaduru plant, kaduru fruit. Here in coconut, the coconut shell is the endocarp of a fruit. The fruit pericarp has three layers: exocarp, mesocarp, and endocarp. This endocarp is very hard in mango and kaduru, and this coconut, this kind of plants are having thick endocarps. They are having sclerates in them. And then pericarp of guava and pear. We can feel that when we eat a pear, we feel that some rough nature. That is due to the presence of sclerate in them. And then seed coat of coffee. It's very thick. That thickness is given by sclerenchyma tissue, that is sclerates. And then second type, fibers. It can be mainly found in xylem tissue and phloem tissue. Xylem fibers and phloem fibers are the fibers present in those tissues. Here you can see these brown color polygonal shaped cells with thick walls are the fibers. And then fibers can be other examples like coconut fiber and also different plant fibers like hemp, agave, and in that type. Here, here it is given jute, and these are fibers. So they contain sclerenchyma cells. And then the function of sclerenchyma cell is, as I mentioned earlier, it provides the mechanical support. It gives the strength to the plant. Normally, as I told you earlier, they have a ring cambium. All the cambia in the vascular bundles connected together to make a ring at the end of the plant growth. When the plant is growing, it becomes a ring. It produces more and more xylem fibers and different xylem cells. There are many cells in a xylem tissue. They are producing more and more. When they are producing more and more, when we are learning xylem tissue, we are learning that most of them are dead cells and they are very strong. They provide the mechanical support. When they are collected in this area, that means inside the stem, it provides a good strength to the plant. That's why we are taking this pith area of a plant as a wood. For our wood purposes, we are taking this middle area of a stem because it contains large amount of xylem vessels, large amount of fibers and xylem vessels like xylem tissues. So they provide in the mechanical support. The fibers in it is a sclerenchyma cell. And then that is all about simple permanent tissues. So, Minindu, now we have come to a point where we have to start a new part. So since it is 8:30 now, do you want to stop? I feel it's better to stop that at that point because we have to start a new part later. What about the things we have discussed up to now? Do you have an clear idea about the simple permanent tissues and main two types of tissues we have learned? They are simple tissues and complex tissues, and before that, meristematic tissues and permanent tissues.
Yeah, miss, I can
Minindo,
hear.
now
Yes,
it's
miss.
8:30.
It's okay, miss, to get bit late. It's okay. If you think
Uh,
it's better to
what should
complete the other part, it's okay. It's okay, miss. We can just do that.
what about your idea? So since you were tired, I'm asking. That's why. Shall we do? I- i- If it is okay, I can continue to the complex tissue part.
Yeah, miss, it's okay. Let's go for it.
Yeah. Okay.
Excuse me, DJ. Mininder, your mom said you are a bit tired and you want to, you want a early break. So it's up to you to decide.
It's
We
okay,
can
sir.
have a fresh session next week. It's up to you to decide whether you want it or whether you move out.
It's okay, sir. If I'm okay with it.
You okay with what?
Uh,
You go ahead, though.
let, let's go ahead.
You sure, right?
Yeah, sure.
No, we are not forcing. That's why. If you are tired, it's no point. Uh,
I'm a bit tired, but it's okay, sir.
all right. Okay. Let's go then. Okay. Go ahead, TJ.
Okay. Okay, so we are going to continue the complex tissue part then. Okay, now we are at the complex tissues. They are xylem tissues and phloem tissues. In this diagram, you can clearly see the, I mentioned this earlier, this area contains different types of tissues and they are performing a particular function. And this area also contains another tissue type, they performing another function. So this middle area, which is at the inner side of the stem, we call it as the xylem tissue. And the tissue area, which is at the outer side of the closer to the outer side of the stem, it is named as phloem tissue. This in between area is the cambium tissue. So this is cambium, which is mentioned here, which is given by this arrow. It is the cambium area. And then we are going to discuss about the first tissue type, xylem tissue. So here, it contains different type of cells. Since it is a complex tissue, it has four types of cells. They are xylem vessel elements, xylem trachea, xylem fibers, and xylem parenchyma. Here, this is the xylem vessel element. It has some pores in it, and here it is somewhat different. This image should be a complete hole here. Normally it has a complete hole, but sometimes they are having some perforations. But normally in this section in grade 11, we are learning that it is having a pore at the middle of the xylem vessel. And then this is a xylem trachea. It is, uh, it is having a tapering end at the end of that. It is having a streamline shape, and also in the same fibers are also having the same shape, but fibers are shorter and narrower than this trachea, which is given here. Strategies are very wider than fibers. And then here, these all three cells are dead cells. They are having lignin thickenings in their walls, in their cellular cell wall, then they are dead cells. But these parenchyma cells, we know it's a live cell, which is in xylem tissues and also in phloem tissues. Then these are the tissues in phloem tissue. These are the cells in phloem tissue. Here you can see this is the main cell we can find in a phloem tissue. It is named as sieve tube element. This upper end or the at the end of each element, sieve tube element, we can see a sieve plate which is having pores in it. And in adjacent to that, there's a cell named as companion cell. It's like a friend to this sieve tube element. Sieve tube element doesn't have a nucleus, so this companion cell helps to these sieve tube elements to carry out its cellular functions. And then it has phloem fibers. This is the phloem fiber. It is a dead cell, as we have learned. It's a sclerenchyma cell. Then phloem parenchyma, they can be seen here. These are the phloem parenchyma cells. The specialty of this companion cell is it has a clear prominent nucleus in it because it has to control both companion cell and also sieve tube element too. So now we are moving to discuss about the differences between the xylem tissue and phloem tissue. The main difference we have to discuss is the difference between the sieve tube element and xylem vessel elements. So it is there. This one is a dead cell. Xylem tissue is a dead cell. Xylem tissue contains this xylem vessel element, it's a dead cell. And then the phloem vessel or the sieve tube element, it's a live cell. But it lacks a nucleus. It doesn't have a nucleus because it has to transport food in it. Therefore, there's no nucleus in it. There should be a space for that, but it's a live cell. And then here in xylem vessel elements, all the cross walls which should be there, but they have completely dissolved. They have completely dissolved and all the xylem vessel elements are connected together. They are stuck on each other to make a continuous tube-like thing. It's just like a transporting water carrying tube. So it's a continuous xylem vessel now. It carries water and mineral. And then here in this phloem tissue, the cross walls are not completely dissolved. They are partially dissolved. We can see the holes, but the wall is there. So sieve tube elements also stack on each other to make a sieve tube or the phloem tissue, the main xylem, main tissue or main cell of the phloem tissue. It is transporting water mixed with mainly food. Glucose is the main product which is transporting along the phloem vessel. We call it as the sieve tube element. And then these are some other differences between these xylem tissues and phloem tissues. Xylem parenchyma is the only living cell in xylem tissue. That is the only living cell. All other cells, that means trachete, xylem vessel element and xylem fibers, they are dead cells. Here in phloem tissues, xylem fiber is the only dead cell. All other cells are alive cells. And in case of xylem tissues, trachete is a spindle-shaped elongated cell. Trachete is a spindle-shaped elongated cell. And in case of phloem tissues, they have companion cells with them, as in the same way like trachea, but they are live cells and they are associated with sieve tube elements. They are having a prominent nucleus to control the functions of the sieve tube element too. And tracheates are broad and longer than fibers. And then here, when we think about the function of the xylem tissue and phloem tissue, xylem tissue transport water and minerals, and phloem tissue transport, here it is, there's a mistake. This should be transport food. It transport food. It transport food. And rather than transporting water and minerals, xylem tissue provides the mechanical support to the stem. So these are the functions of those plant, complex plant tissues. Okay, Minindu, we are moving to the animal tissues. Is there any question you want to ask from me about these xylem tissues and phloem tissues and their function and all? Did you understand these things?
Yes, miss, I'm clear with it.
Okay. Then we are moving to discuss about the animal tissues. Animal tissues. Here there are four main types of animal tissues in a body. They are epithelial tissue, connective tissue, muscle tissue, and nervous tissue. Here, at first we are going to discuss about epithelial tissue. It is the tissue which lines the internal and external surfaces of the body. The most common feature of that is it has a basement membrane and cells are top of that. There may be one cell layer or many cell layers in an epithelial tissue. It can be different, and also the shape of these cells can be different. This is somewhat cubic shape, but these are some flat cells. So shape can be different, number of cell layers can be changed. And cells are tightly packed. That is another special feature. There's no holes in between them. There's no spaces in between them. They are tightly packed and they are on a basement membrane. We cannot be seen fibers in these epithelial tissues. And then it has a nerve supply and there's no blood supply. When we have some type of a scratch or something on our body, we can see that a white color membrane like thing is removing, but there's no blood coming out, but we feel the pain. The reason for that is it has a nerve supply to feel, but there's no blood supply. And then we are going to discuss about the functions of this epithelium tissue. Normally these epithelium tissues can be found in the internal and all the external surfaces of the body. The main epithelial tissue we can find is which is on our skin. So it's a epithelial tissue contain large number of cell layers. Many cell layers are there. But our body has some places which are having single cell layer epithelial tissues. So the first function of this epithelial tissue is to lining of the surfaces and protect the internal organs. They protect the internal organs from pressure, friction, and microbes. They prevent the entry of microbes, and if there's an abrasion, they provide that friction, and they prevent that friction. And also if there's a pressure between organs, they prevent the harm which can be caused to those internal organs. And then they perform an absorptive function in a digestive tract. That means in our large intestine or in our small intestine, they can absorb different material in. Mostly in our digestive system, they absorb nutrients. In large intestine, they are absorbing water. So that process is happen via a single cell layer of epithelial tissue. Here you can see in this image, this red color cells with some few small projections on it named as microvilli is the epithelial tissue in our digestive tract. Here in this area, this is the middle area of the digestive tract. The food, digesting food is flowing in that area. All the substances, some glucose and different minerals and all the nutrients are absorbing to capillaries and also to lacteals. Lacteals absorb the mostly lipid things and capillaries absorb all the other things like medicines, what we have taken in, and other nutrients. All the things are absorbed by these capillaries. So it's the absorptive function. And then perception of stimuli. In nose, skin, tongue, in nose we can feel the smell that is uh, carried out by the cells in our epithelial tissue. Nerve ends are in the nostrils, and also there are some nerve ends in our skin, tongue. In that way, they can perceive the different stimuli, smell, uh, temperature, pain, and the flavor we can perceive with that. And then the fourth function is secretory function. They can secrete different things. The most common example is our respiratory tract. They have some cells to secrete mucus. They secrete mucus and trap all the pathogens and trap all the dust we entered into our respiratory tract. They trap them and spilled out. So it provide a secretory function to secrete the mucus. All of that is done by the cells in the epithelia of those area. And then last function is the filtering function. Normally, which is done, this is done in the nephron of a kidney. It has a special part named as Bowman capsule. In this Bowman's capsule, you can see here, this red color part is a blood capillary, complex of a network of blood capillaries. And some substances are filtering out to this Bowman capsule. It's just like a cup-like structure. All the unwanted material, maybe water, some extra water, what we have taken, and there may be some different medicines, and there may be mainly our excretory substances like urea, uric acid, and all the things. They are not needed for our body. They are filtered out to release out with urine, and that is the filtering function. There's a single cell layer of epithelium tissue in this Bowman capsule and also on these blood capillaries. And then we are moving to discuss about the connective tissues. That epithelial tissue carry out the function of lining up the surfaces. These connective tissues are carrying out the connection to maintain carrying out the function of maintaining the connection between different tissues and organs. The most common example for the connective tissue is blood. So as we can take this as an example, it provides a good connection between all of the organ in our body. As an example, it takes oxygen from our lungs. Our lungs have alveoli. It's a special structure where the air exchange occur. These lungs provide oxygen to our blood, and blood release carbon dioxide where there was there where in that blood, and carbon dioxide is removed out, oxygen is taken in. Then what this blood do is it goes all along the body and provide this oxygen to our cells. Each and every organ is connected with blood tissue, so it's a connective tissue. And not only blood, there are some other tissues too: bones, cartilage, and adipose tissue. That is the tissue between our skin, which is having a store of lipid. So that adipose tissue, that is also named as, sometimes named as the, that upper below layer is named as the hypodermal fat layer. That adipose tissue is also a connective tissue. It provides the connection between our skin and inner area. And then it has some different, some special features. There are different types of cells and fibers can be found. They are present in a connective tissue. Different types of cells and fibers. And then these cells and fibers are embedded in a matrix. Normally at the epithelial tissue, we saw that they are in a basement membrane. They are on a basement membrane. They are not embedded anywhere. But in case of these connective tissues, the cells and fibers are embedded in a matrix. Did you understand that?
Yes, miss.
Yeah, they are embedded in a matrix. Then they have both nerve supply and blood supply. Both nerve supply and blood supply is there. Those are the special features of them. The most special features are these two. Different types of cells and fibers are present. Cells and fibers are embedded in a matrix. Then we are going to discuss about this connective tissue, which is named as blood. So it's a special connective tissue because it has a fluid matrix. Normally, in case of these bones, cartilage, and adipose tissue, they are not just like liquid things. They are very strong, hard material. Bones are very hard, we know that. Cartilage is also hard, but not much like bones. And also adipose tissue, it's also not a liquid, but this blood having a fluid matrix, which is named as blood plasma. It's a fluid matrix. All the cells are embedded in this fluid matrix. There are different types of cells in this blood tissue. There are red blood cells, those are named as erythrocytes too, and white blood cells, those are named as leukocytes too. And then finally, this is not a cell type, these are just cellular fragments. They are named as platelets. They are cellular fragments. These are complete cells. And then fiber, there's another special feature too. Normally in bones, cartilage, and adipose tissue, we can find fibers easily. But in this blood tissue, we cannot find fibers every time. They are not available all the time, but we can see at one incident, that is blood clotting. During the blood clotting, when we have a wound, these fibers are formed. These are normally named as prothrombin and fibrinogen. They are two protein fibers. These fibers are coming here and they are formed here and make a net-like structure to prevent the flow of blood. Then that is the blood clot. These fibers can be seen only at that event. That is the blood clotting. And then the functions of blood tissue. We are going to discuss what are the functions of this special connective tissue. Since this is a connective tissue, it has a connective function actually. That is the transportation of different material throughout our body. There may be respiratory gases, and also there may be nutrient, hormones, excretory matter. All the things are transported throughout our body. Respiratory gases are transported between all the cells and lungs. Lungs gives the oxygen and it comes to the heart, and then heart pump all the blood which contain oxygenated blood to the other cells of the body. And then hormones, they are produced in some different glands, only few glands in our body. But those hormones are transported through our body to carry out different functions. As an example, uh, growth hormone is produced in a gland which is producing growth hormone, but that hormone is acting here and there in our body to grow us. So it is transported by this blood tissue. And excretory matter also transported to the kidney to remove it out of the body. And then next function of blood tissue is the protection. It gives us the protection from the pathogens. These pathogens are destroyed normally by phagocytes, by phagocytosis. Here it should be come with this one. These phagocytes carry out the process of destroying pathogens. When a bacterium or a virus enter into our blood tissue, what these phagocytes do it, phagocyte is actually a white blood cells. You can see here. They are neutrophil, basophil, and lymphocyte, monocyte. All of these things are white blood cells. You are learning these things in lesson number 6. All the tissue, all the information about blood tissue and blood circulatory system too. So these white blood cells are carrying out the function of destroying pathogens. These phagocytes engulf the bacteria and destroy them. That process is known as phagocytosis. And then there's another method. Rather than engulfing and destroying by phagocytosis, they produce some weapons named as antibiotics. So the another function of this blood is to produce antibiotics, to destroy pathogens. And then as the third function, they maintain the homeostasis in our body. As an example, our blood temperature can be controlled. Our blood glucose level can be controlled. And there are many this kind of functions in our blood tissue. They are controlling our body. When the body temperature is high, our blood tissue try to control our body temperature. As an example, when we are having a bath and we feel there are some differences in our fingers, they are just like the shape of the fingers are changing due to the absence of the blood supply to our finger ends, fingertips. So that is for the control of the temperature of our body. And also there are many, but that is the main function as the homeostasis. So these are the function of blood tissue. Later on, we are moving to discuss about the muscle tissue. Here, I hope now you can have a better idea about connective tissue and the blood tissue as an example. And then we are moving to muscle tissues. There are 3 types of muscle tissues. All of these muscle tissues are performing a special common function. That is, their contraction and relaxation ability to make different movements in our body. Uh, these three types of muscle cells are found in three different places, and they are moving in three different patterns to do different functions in our body. Sometimes this smooth muscle tissue, it is in a particular place, it moves slowly. And as an example, this skeletal muscle tissue, it is having a fast movement ability because it is in our legs and our face and also in our hands too. So this cardiac muscle tissue, it has a unique moving pattern. It is having a rhythmic moving pattern because it's in our heart. So in this way, these three are performing three different movements in our body. So here we are going to look at their features at first before talking about their functions, their functions and their differences in their control. Here we are going to discuss about their features. In case of smooth muscle tissue, these smooth muscle cells are spindle shaped. They are having a tapering ends at the end of the cell, and they are not branched, they are unbranched, just a single cell, and uninucleated. There's only one cell at the middle of the cell, uninucleated. And then when it comes to skeletal muscle tissue, they are long and cylindrical cells, long cylindrical cells. They are unbranched, as in the same way like smooth muscle tissue, they are unbranched, but they are longer than smooth muscle tissues. They are having many nuclei. That is the difference from smooth muscle tissue. There are many nuclei. And another special feature of these nuclei is they are at the periphery of the cell. They are they are around the cell, not at the middle of the cell as in the smooth muscle tissue or in the cardiac muscle tissue. They are at the periphery of the smooth skeletal muscle fiber, skeletal muscle cell. And then another special feature they have is these striations. There are some cross lines in their structure. They are named as striations. These striations can be found in both skeletal muscle tissue and cardiac muscle tissue. These are the striations. That's a common feature of both skeletal muscle tissue and cardiac muscle tissue. And, and then I am moving to tell you about this cardiac muscle tissue. When we look at this skeletal muscle tissue, it is long and cylindrical, but it is unbranched. But here, this cardiac muscle tissue is branched. This is short and cylindrical and branched. And also especially it is uninucleated as in the same way like smooth muscle tissue. It is at the middle. But once we look at this cardiac muscle tissue, we feel like that they are long, but it is not like that. These cells are separated by a special area named as intercalated disc that separates the cells. So we can think that the cells are short. Here, this is a one single cell of cardiac muscle tissue. It contains one nuclei and it is branched, has many striations on it, and it is cylindrical. Cross section is cylindrical, circular in shape. So these are the features of three types of muscle tissues. Then we are moving to discuss about their functions. Actually, what is the control? How they are controlled? How they are moving? We have to remember that. And do they fatigue? And where they can be found? We have to know these things. So here we are going to discuss about that. And here it is mentioned that the muscle tissue is a tissue with blood supply and nerve supply. So we don't have to tell that actually. We know that when there's a cut, we know that when there's a wound, there's a pain of a muscle. When there's a wound, we have a blood release. So they have blood supply and nerve supply, obviously. So here, in case of smooth muscle tissue and skeletal muscle tissue and cardiac muscle tissues, their control is important. This skeletal muscle tissue can be controlled by our brain. If we want to move our hand, we can do that. That means they are voluntary control. Voluntary means by the knowing of the brain. That means by the control of the brain. It can be controlled by brain. But other two types are not like that. They are involuntarily controlled. That means smooth muscle tissues are in the lining of internal organs most of the time. As an example, our stomach, our bladder, and also our intestine walls, they contain smooth muscle tissues. So we cannot control them. If we want to control the movement of our stomach, it cannot be done. It moves itself. It moves by itself without the knowing of our brain. Our brain is not involving to their movements, involuntarily controlled. And then cardiac muscle tissue, it's mostly, it is obviously it is controlled by itself. We cannot control the heart movements. They are moving by themselves. Those muscle cells are moving by themselves. So they are involuntarily controlled. And then when we look at the speed of their movement, smooth muscle tissue moves very slowly. Sometimes they are just like rhythmic movements in our stomach. We feel hungry when there's no food in our stomach, because our stomach is moving very slowly to digest, for the mechanical digestion actually, to break down food into small part. When we eat some food, it is broken down by our teeth at first, and then in our stomach, they are breaking down into small part further. So those slow movements are to the digestion of food. When there's no food, we feel the movements of that stomach. That we call that as hunger. We feel a particular pain called hunger due to those movements. And then that's a slow movement. In case of skeletal muscle tissue, we know that they are doing very fast movement. When we want to run, we can run because our skeletal muscle tissues can do fast movement. And then due to that feature, they are having many mitochondria. Because to make fast movements, they should have more energy. To produce more energy, they should have many mitochondria in those cells to do cellular respiration. So that is the reason. And then in case of cardiac muscle tissue, they are having rhythmic movement. The heartbeat, we call it as the heartbeat. It has a particular rhythm. It perform rhythmic movements. And then I'm coming to smooth muscle tissue again to look whether they are becoming fatigued. Smooth muscle tissues become fatigued very rarely, but they don't become fatigued very quickly. But in case of skeletal muscle tissue, they become fatigued quickly. That's why we feel tired. We can't move our body by just after running for a long time, or just after walking for a long time, or maybe stepping up in a staircase or somewhere. We feel tired. That's due to the becoming fatigue feature of this skeletal muscle tissue. And then cardiac muscle tissue, it never become fatigue. If it become fatigue, we have to end the life, because heart is going to be stopped if it is going to be fatigued. Cardiac muscle tissue never become fatigued. And then smooth muscle tissues can be found in the walls of the digestive tract, uterus, bladder, and blood vessels. They can increase their volume. That means they can show dilations. They can increase their volume due to the uh, relaxation of these smooth muscle tissues. Then they can contract due to the contractions of these smooth muscle tissues. Volume can be increased and decreased. The bladder can be increase its volume. There's high amount of urine in it. Then it becomes small due to the contraction of smooth muscle tissue later. And then skeletal muscle tissue can be found in biceps, triceps muscles in our hands, and also muscles in legs, and also in our face. Our facial muscles also skeletal muscles, because we can move by their knowing, by the control. They are under the control of our brain. And then this cardiac muscle tissue can only be found in our heart. That means invertebrate heart. This is all about muscle tissues. Vinidul, did you understand these things?
Yes, miss.
Yes. Okay, shall I move to the next part? It's about nervous tissue. That's the last part actually.
Okay, miss.
Okay. So this is about nervous tissue. And then that is the last type of animal tissue we are going to discuss today. So in nervous tissue, the structural unit is the neuron. Here in muscle tissue, they are smooth muscle cells, skeletal muscle cells, and cardiac muscle cells. They are the building blocks of all the tissues. And in case of blood tissue, there are different cells, not only one single cell. And here, in case of nervous tissue, it has a structural unit. The special structural unit, this is named as a neuron. Neuron has two parts. Neuron is the cell which create the nervous tissue in our body. It has main two parts. When we take a typical neuron like this, it has main two parts. This is the cell body, which is mentioned here is the cell body. This whole part is the cell body. And then it has some nerve fibers like this. There are a few fibers in this area, and there's a long fiber in this region. So these are named as the nerve fibers. Then cell body and nerve fibers. These are the main two parts of a neuron. And then the cell body contains different cellular organelle, mainly a large nucleus, because this is a very important tissue in our body. It is very special. It is having a very special structure. It has a large nucleus and it has some special shapes to transmit nerve impulses through that. So as the nerve fibers, there are two types of fibers, nerve fibers. One is axon. It is the longer one. It is the longer nerve fiber, and there are some different short nerve fibers named as dendrites. These are the dendrites, those which have branch in this way, and axon also branch at the end of it. We call these as axon, branches of this axon, and at the end of this there are some round color objects. We call them as axon terminals, or there's a special name. They are synapses vesicles. Synapses vesicles. Synapses. That is the special name. Okay, synapses vesicles at the end of this axon terminal. And then we have some special feature here. We have to learn about these objects. These yellow color parts are named as myelin sheaths, or the single part of it, single object of that is named as Schwann cell. But it is not given in our book, but just remember that name. That is just like a cell. It is known as Schwann cell. And at the top of this cell, that area is named as myelin sheath. In between these myelin sheaths, we can see that nodes of Ranvier. That is just a interrupted place in between those myelin sheath. That is named as nodes of Ranvier. These myelin sheaths are very important for the transmission of nerve impulses, because it increases the rate of transmission of nerve impulses. Because rather than walking in a way, it is better to jump from one place to another. That means it increases our speed, like we are walking on a railway track. It's just like that. Now impulses are transmitting in a faster way due to the presence of these myelin sheaths. It accelerates the rate of transmission of nerve impulses. Then the functions of nervous tissue. As we know, it transmits the information to different, here it should be not in this place. Here these are not effectors. It should be receptors. It should be under the second part. It's a mistake. They are receptors. We know that there are 6 receptors. We have eye, ear, nose, and skin, tongue. These are the receptors. So they perceive some stimuli. Those perception stimuli are transmitted along the different nerves to our nervous system, central nervous system. Sometimes these nerve impulses, or the information which are perceived from the external environment, are transmitted along the nerves to another nerve. And then sometimes they are transmitted from the nervous system to an effector. So mostly these are the receptors in this part. Effectors are our muscles. Our muscles act as effectors. As an example, if we feel that our finger is going to be burned with a candle or something, we just feel the heat. So our skin get that information. Our skin perceive that stimuli of heat or the temperature and transmit that information from that particular receptor to our central nervous system along a particular neuron. There are 3 types of neuron in our body. I'll tell you that in next slide. Along one neuron, that information is sent to our brain or the central nervous system. Then our brain identify there's something danger, and it gives a signal to move our hand away. Then our muscles in our hand are the effectors. They get the information from the brain and it make a movement to take our hand away. So that's how nerve impulses are moving. To do this transmission of information, we have three types of neurons. As I told you, this sensory neuron is the one which takes the signals in. It is mostly connected with our, it is always connected with our receptors. They are on our receptors. So they take the information and transmit along that to the interneuron. So interneuron take the information from the sensory neuron and transmit it to the motor neuron. Interneuron is at the middle of our body, middle of our nervous system, that is the central nervous system. Then it gives a particular signal to move our hand or whatever. It is given to a effector. An effector is mostly a muscle. This is mostly a muscle. So we call this as the effector. It gives an effect. It is effector. So this is the way of nerve, the transmission of nerve impulses. This is the sensory neuron and interneuron and motor neuron depending on their different functions. And here I couldn't tell you that normally transmission of nerve impulses occur from cell body to the axons. So the function of these dendrites is to transmit nerve impulses towards the cell body. It transmits nerve impulses towards the cell body. That means it takes the nerve impulses to neuron and then it is sent to the axon. So axon transmit nerve impulses away from the cell body. So these red color arrows are giving the direction of the transmission of nerve impulses. So Minidu, do you understand these things? Can you understand?
Yes, miss, I can understand.
Yeah. Okay. So today I did it bit faster because we have only a few weeks. That means just about 34, 35 weeks to end this year. So we have 15 lessons. Since this is a small lesson, I did it bit fast. What do you think about the speed of the lesson? Is there any problem with that? Is there any suggestion to control the speed or something? What do you want to tell me about that? Is there something?
Miss, it's okay with this speed, but if I have
Yeah,
problem, I will ask miss.
yeah, okay. I think then today you will have some problem with uh, not having a TO to write like that. You have to listen all the way. So it's somewhat comfortable. I know that. But so next day we will uh, continue with the tune. Then it will be easy for you to join with the class in the next day. Okay.
Okay, miss.
Okay. Minidu, I'm going to upload a question paper from that lesson. So you can download it and you can answer it and you can upload your answers. Okay.
Okay, miss.
Okay. Then we are going to stop. So I'm going to stop the class. So good night, Minidu.
Good night, miss.
Okay.
Thank you, Miss.
Okay, you are welcome. If you have any problem, you can call. There's a number you can call. Shall I give it to you?
Yeah, Miss, sure.
Yeah. Okay, it's 076. Can you
Just
write?
wait a second.
Okay.
Okay, Miss.
Okay, 076
Okay, Miss.
571
Uh-huh.
0930. That is the number. If you have something to tell about the class or whatever, about the platform, maybe you may have uh, some problems when you are using the platform, or when you face some kind of a situation, you can give a call to this and ask for a solution. Okay, so
Okay,
I'm
Miss.
going to stop the class. Good night again. Bye.
Bye, Miss. Good.